Welcome to the daily update for April 15th. And like usual, I'm going to just kind of walk you through what the expectations are and what you need to do for today's lesson. So hopefully by Wednesday, you had finished between Monday and Wednesday the case study for the patient Michael. And you should have written up at the end a claim of what you think Michael had with your evidence and reasoning for it. Today, what you're going to do is it's going to piggyback on that same idea, and we're going to go with a little bit more reinforcement. Usually, I like to talk about the answers before the reinforcement, but I still have students that haven't turned it in. So I really need you guys to please make sure that you are turning in your items. I can't give feedback until I get everyone's work. Now, I know a couple of you have emailed me separately, and that's fine. But there are still people that haven't contacted me that haven't turned in materials, and that's what I'm concerned about. So I went into week 14. Monday and Tuesday were the genetics case study, or Monday and Wednesday. I'm so not used to the staggered days. For Friday, it says complete the reinforcement assignment below. It's similar to the last case study. And then any extra time, I'm giving you these quizzes back. Some people have played them. I've seen that. The Quizlets and the Central Dogma Interactive. On Monday, it said that we were going to, on Wednesday, I said that we were going to do one more mutation lesson, which is this one, and then start a research project. I'm actually going to push it a couple of days off because on Monday, before we start doing the research, I want to talk to you about how all these traits that the proteins express, how this DNA is actually passed on to offspring, because we've talked a little bit about how DNA can replicate and how cells can divide through mitosis. And that's one form of reproduction for asexual animals such as bacteria, but we haven't talked about sexual reproduction and how that's a little bit more different. But we're going to spend at least a day or two, Monday and Wednesday, doing that. And then we'll start getting into your research project, okay? I just want to explain a couple more details first. But for today's reinforcement lesson, to piggyback on again, it's a Google Doc, so you can write directly on it when you open it. Again, we're going to be given DNA sequences, okay? These are going to be partial sequences. Now, instead of just... And last time our two strands were normal and an abnormal. And we were told that, the patients. And this one, it's going to be humans and cows. So it's not necessarily normal versus someone else's. It's two different organisms themselves. Okay? But you're going to kind of do something similar. You're going to use the DNA sequence to make a complementary RNA strand from both the human and cow and write directly below it. I have a codon chart here, because then you're going to write the correct amino acid in the row below here. So the human DNA is first, and so you will transcribe into your mRNA strand, each of these, and then you'll take your mRNA, use this codon chart to determine the correct amino acid sequence. So that's kind of like, I think it was part 2 on the patient Michael case study that we did. Now, one of the things that I wanted to tell you is that this is a sample sequence for a protein, a gene that makes insulin. So insulin helps regulate our blood sugar. If you know anyone that's diabetic, it's usually because there is a defect in their gene that produces insulin or uptakes it, or there's also other molecules involved called like glucagon as another one, glucose. There's other enzymes involved too, but there's usually a gene that is somewhere involved in connection with insulin. Without insulin, a person can't digest sugars the same ways others people can, okay? So we're going to look at humans, and this codes for insulin. This is not the entire amino acid sequence. The entire amino acid sequence for this gene is 51 amino acids, which if you times it by 3 bases, 3 codons, you're looking at 150 letters, 150 nucleotide bases in that sequence. Then you're going to do the same thing with a cow. They both have a gene that regulates the production of insulin. Okay. Now, just like you compare the normal to the patients in the previous case study, you're going to compare these two. Okay, you're going to circle any ones that's different, highlight any amino acids that are, circle any single bases that are different and highlight any amino acids that are yellow. And then you're going to answer questions 3 and 4. Okay. Remember in the screencast and in the video that we talked about how some of our amino acids have more than one codon. It could have up to 4 codons that code for the same amino acid because there's only 20 amino acids, but there's 64 combinations of codons. That's what this is asking for. Okay, so if you're looking at leucine, you need to identify all the possible codons that could code for that. After that, we're going to look at the mutations. Mutations, there's something that happened that's different in the DNA. And there's different types. There's point mutations, there's a frame shift mutation, and a nonsense mutation. And so you're going to read through these and then answer the questions. And then what's going to happen is you're going to start, instead of just comparing a patient to a normal functioning person, a human's DNA to another human's DNA, like we were doing in the previous exercise, you're going to compare a human to a cow. What we're going to do is these sequences and these changes in sequences can also be very useful when we're trying to determine how closely different organisms are to each other, evolutionarily speaking. We kind of looked at it when we did some in the Africa case study when we were looking at the DNA sequences of different lions. I think it was different lions in that case. You've seen different sequences and had to look at differences to figure out, what I think what place you were in for our seals, for our seal pods too, when we did our homeostasis unit. And so it's the same idea here. The more differences, the further apart they are evolutionarily speaking. So you're going to be given 4, and you're going to have to figure out which 2 are most closely related. And then you're going to answer these wrap-up questions, okay? Finally, the last thing is sort of your assessment piece. Construct an explanation based on evidence using data from up above for how the structure of DNA determines the structures of protein which carry out the essential functions of life through the system of specialized cells. Use the image as a guide. And as I am noticing right here, and I don't know if it's because there's a glitch, but the whole picture's not showing up. So let me see if I can adjust that right now. Oh, and here's my daughter, Charlotte, coming to join us. There we go. You know what I think I might do is just put this onto the next page. There we go. So that way we can still keep it bigger. There we go. So you're going to use this image as a guide. So the DNA goes to the RNA, translated to the amino acid sequence, which then terminates and folds up into your protein. And so you're going to kind of explain how this leads to this. Okay? So let me know if you have any questions. And Monday, we will have an ed puzzle and a brief screencast lecture. I might also do a Google Meet if people would rather do a real life lecture. I'll have a screencast of the lecture, and I'll also have a Google Meet. So I will let you know what time the Google Meet is. So in case you want to just do it in real time and ask questions as we go through, you can do that. But that will be on Monday, okay? Anyways, if I don't talk to you, have a good weekend, and I will see you.